Mice rely on their vision for most of the important aspects of their complex lives. Now researchers have gained new insights of mouse vision by linking behavior and retinal function.
My name is Jason Kerr. I run a lab, BBO, which stands for Behavior and Brain Organisation. We are interested in how mammals use their vision to make decisions and the underlying neural circuitry. You're about to hear from Paul and Carl about their new exciting paper in which they could see what the animal was seeing as it ran around after prey.
We've recently published a paper in which we developed new tools and techniques to be able to track a mouse's eyes as it hunts its prey. And from this we were able to reconstruct what the mouse saw from its perspective for several reasons. First, their eye movements are tightly bound to their head movements. So when they explore an environment, when they hunt, the eye movements counteract the head movements. Secondly, like many prey animals, their eyes point out to the side, and each eye has a wide field of view. This has the advantage that they have a panoramic view of their surroundings. Being able to stabilize your large panoramic field of view is a great advantage if you're a small mammal trying to avoid being eaten by predators. But if you don't have any real voluntary control of your eye movements in your head, like humans do, then how do you use your eyes to search for prey? In other words, if your eye movements counter your head movements and you have a large panoramic field of view, which part of your retina do you use when you're trying to detect and then track a small moving, fast hopping object that's fleeing away for its life, and to what advantage? During the experiment we needed to capture several aspects of the mouse's behaviour, but when mice hunt they make use of multiple senses. They can hear their prey, they can smell their prey, and when they get close enough they can touch their prey using their whiskers and their paws. But what we were really interested in was how do they use vision both to detect and then to track their prey. To mask the smell of the one cricket in the arena, we used a fan to blow cricket scent from dozens of crickets into the arena. To mask any sounds from the cricket, we played white noise through speakers in the walls of the arena. And to avoid the component of touch, we analysed the recordings until just before the point the mouse came into contact with the cricket. The mouse would have to actively detect, track, and finally capture the cricket. And the cricket could escape, and the mouse would have to repeat this process multiple times before it finally caught its meal. To capture the mouse's head and eye movements, during the experiment the mouse wore a small, light, detachable eye camera and head position tracking system that we'd previously developed. And in order to capture the 3D position of the mouse's head and the head rotations, together with the 3D position of the crickets in the arena, we had a suite of cameras above the arena to capture all the action from different angles. The next step was to recreate the view that the animal would have seen. To do this, Paul and other members of the lab recreated the mouse eye views as it hunted down the cricket.
The goal of our analysis was to reconstruct the mouse point of view as exactly as possible. To achieve this, we combined head and eye tracking Carl captured during the experiments with a digital scan of the complete room, accurate to the millimeter. The resulting model allowed me to compute the animal's viewpoint, the distances, the object motion, and the relative 3D positions directly from the data, something which is not possible with a simple video.
Using the mouse eye views, we saw that mice hold their prey in a small spot on the eye, and this region is sampled by a retinal area that is thought to aid tracking objects. How did they do this when their eyes counter their head movements and they can't really voluntarily move their visual fields? What mice do is when they detect a cricket in their large visual field, they run directly towards the cricket. If the cricket jumps away, they repeat this and run directly towards the cricket again. It turns out that this area is the area of least motion-induced blur as the animal runs forward. So any object within this area is likely to be clearer and less affected by motion blur as the animal runs forward. We can extend our research into the retinal region we identified and examine which brain regions this connects with. And then using our new head-mounted 3-photon microscopes, we can start to unravel the neural mechanisms underlying this visual behavior. And finally, we can use these techniques to study other freely moving animals. to see what they see as they perform a behaviour.